suicide does not fit so well into the concept of survival of the fittest. So aside from that being quite a thought problem, as a neuroscientist, I'm interested in studying how the brain changes during depression in order to find better antidepressant treatments. And specifically, I'm studying structural changes. So it's commonly thought of that there's like a chemical imbalance in the brain during depression, which is more of a functional hypothesis. But I'm an anatomist. I look at structure. And this is interesting, and it's one of the reasons that it drew me into Convergence, is because I'm looking for a visual structural entity which resembles depression. I'm trying to see it. And that's sort of what I've been focused on. It's a great interest of mine. I've tried to get it across with the paintings, which I will now try and work from. So I work on a cell called astrocytes. They're a star-shaped cell, which I painted here. They're 3,500 times larger here than in the brain. It's all to scale. And they're a star-shaped brain cell in the brain, which they clear debris, and they give nutrients to cells which function the neurons, and they also remove waste, so they're the cleaners of the brain. And what's interesting is that in depression, these seem to get smaller. So there's a structural change in depression that seems to happen. And I've shown them here contacting blood vessels, because another thing which we're finding in depression is that there are vascular changes. And because these cells are getting smaller, they might be contacting fewer vessels, and they might be contacting fewer cells. And so, in summary, there might be a buildup of waste in the brain because these cells are getting smaller, they're not contacting blood vessels, and so their function is impaired. But at a visual level, we can visualize the structure. The final thing that we need to know about the cell is that these cells can aggregate together and form something called a glial scar at sites of injury. And the proteins they use to do this are not found, they're found at a lower level in depression. So we think that there's a decreased function of these cells to respond to injury, and this might also be what might be leading to depressed mood. So it's the visualizing the size, the connection of these cells with the blood vessels, and also their ability to respond to changes in the brain might give us an insight into what is happening in depression. It might give us a real view of what's going on. This final painting, I'd like to end. I realize I have the whole speech up there. I'm kind of improvising from. But the final piece here is a reflection on the socio-cultural aspect of the work. It kind of formulates my own kind of view, not really representative of the lab. That what's happening here is that we're now learning to associate mental states with cellular shape. Originally, there have been always social institutions which govern the definition of what is mental health. And in the past, that has been religious institutions, and then it has been psychoanalysis through the use of ink blots. Now we're looking at the cell. And it's just been through the process of societies, Western societies becoming more secularized, more empirical. We have now moved from thinking that it's that sorrow, depression is this, through the religious institutions, it used to be sin of the spirit. The psychoanalysts used to think that it was repressed instinct. So it's a mind. It was from spirit of the religion to the mind of the psychoanalyst. Now we're looking at the brain. So we've come a long way in the last 2 centuries in changing our definition of mental health. It's changed the way that we've developed how we respond to it as well. And with that, I'd like to close. We're anatomists. We are at the forefront of what we're now calling mental health. And so we're in the best position to find a new way to treat depression. Thank you.